cells of your body is a large organelle called the nucleus. The nucleus is responsible for storing genetic information on chromosomes, which are thread-like structures made of DNA and protein. Every living organism has a specific number of chromosomes in their nucleus. This is called the chromosome number for the species. Humans, for example, have a chromosome number of 46. Therefore, each cell in the human body should have 46 chromosomes in its nucleus. Cats have 38 chromosomes, while a mosquito has a chromosome number of 6. A chicken has a chromosome number of 78, while a pineapple has a chromosome number of 50. Chromosomes can be organized into homologous pairs. Remember that homologous chromosomes have the same size and shape and carry similar genetic information. The 46 chromosomes found in human cells, for instance, can be organized into 23 homologous pairs. One of these pairs of chromosomes are sex chromosomes that determine if an organism is male or female. In humans and many other animals, there are two types of sex chromosome, the X chromosome and the Y chromosome. If an individual has two X chromosomes, it will be female. Males, on the other hand, have one X chromosome and one Y chromosome. Let's take a closer look at the structure of chromosomes and the molecules that compose them. If you unravel a chromosome, what you find is that it's made up of a long strand of deoxyribonucleic acid, or DNA, that gets coiled around proteins called histones. The DNA strand by itself looks like a twisted-up ladder. This spiraled ladder shape is called a double helix. If we dissect the DNA molecule even further, we see it's made up of smaller subunits called nucleotides. Nucleotides are made up of a phosphate group, a sugar molecule called deoxyribose, and a nitrogenous base. The sugar and phosphate sit on the outside of the helix and compose the backbone of the DNA molecule. The nitrogenous bases form the rungs of the ladder in the middle. There are 4 different nitrogenous bases that are found in the nucleotides of DNA: adenine, guanine, cytosine, and thymine. Adenine and guanine both have a double ring structure and are known as purines. Cytosine and thymine, meanwhile, have only a single ring and are called pyrimidines. A strand of DNA may have hundreds of thousands of nitrogenous bases lined up in the center of the molecule. The specific order in which the nitrogenous bases appear is an organism's genetic code. The genetic code determines which traits the organism will possess. Let's do a quick recap of the DNA structure. On the outside, we have the chain of deoxyribose sugar and phosphate. Sticking out in the middle are the nitrogenous bases: adenine, guanine, cytosine, and thymine. The bases can appear in any order and can even repeat. The order of the nitrogenous bases is the genetic code that determines an organism's traits. What we see here is just half of the DNA molecule. DNA is double-stranded, so we have another sugar-phosphate chain on the opposite side with nitrogenous bases pointing inward. This is the complementary strand of DNA, and the bases in the center must complement or pair up properly with the bases on the other strand. Nitrogenous bases have very strict rules for pairing. Adenine and thymine always pair together, while cytosine and guanine always pair together. Hydrogen bonds hold the bases together in the center. Let's practice by creating the complementary strand to this DNA sequence. The first letter in the top is C. C stands for cytosine. Cytosine always pairs with guanine, so on the complementary strand we put a G for guanine. The next letter is A for adenine. Adenine always pairs with thymine, so on the complementary strand we'll write a T for thymine. Guanine pairs with cytosine. So on the complementary strand, we write C. Thymine pairs with adenine. Adenine pairs with thymine. Guanine pairs with cytosine. And again, guanine pairs with cytosine. Notice that once we complete the complementary strand, there will be equal amounts of adenine and thymine because of the pairing rules. Count and see. If you count up the number of adenines, or A's, on this strand, you see 3. Similarly, if you count the thymines up, you see 3. 3 thymines and 3 adenines. There are equal amounts because of the pairing rules. There will also be equal amounts of guanine and cytosine because these two pair together. Counting up the cytosine, or C's, we see 4 cytosine. We also see 4 guanine. Now that you are familiar with the structure of DNA, let's take a look at how the molecule copies itself. The process of copying a DNA molecule is called DNA replication. DNA replication occurs every time a cell divides so that each new cell can have a copy of the DNA. The first step in DNA replication involves an enzyme called DNA helicase. DNA helicase works to unzip the DNA helix. When the helix is unwound, the two strands separate and the genetic code is exposed. A second enzyme called DNA polymerase reads the exposed nitrogenous bases and adds new nucleotides according to the base pairing rules. Adenine binds to thymine, and guanine binds to cytosine. In this way, 2 molecules of DNA are formed, each possessing 1 original strand of DNA and 1 new strand of DNA. This is referred to as semi-conservative replication. The process of DNA replication occurs at multiple sites along a DNA strand in order to speed up the copying process. If replication began at the beginning of a strand and progressed in one direction all the way to the end, it would take 33 hours to copy a single strand of DNA. Ain't nobody got time for that. The fact that DNA replication occurs at multiple replication forks along the DNA strand allows DNA replication to occur at a much more rapid pace, saving valuable time for the organism. DNA replication occurs mostly without errors. As DNA polymerase adds new nucleotides to the chain, it also proofreads to prevent mismatched pairs. If there is an error, DNA polymerase can go back and fix it. Every once in a while, polymerase does not catch an error, and the mismatched pair remains. This change in the genetic code is called a mutation. Mutations can be problematic to the organisms. Mutations can also sometimes be beneficial. Other times, mutations don't seem to have any effect whatsoever to the survival of an organism. We'll discuss mutations and how they can affect an organism in a later lesson.